Hello and welcome to Toddcast 53. This will be the last of the free radical podcast. This will probably end up being the most important one for your trajectory in science classes. As far as OChem goes, maybe not the most important, but as far as OChem, biochem, physiology, cell bio, whatever class you should go into that also address antioxidants or free radicals, this will be probably the most important. As a chem major, this is by far the most important if you happen to fall in that category. So this continues from the what we've done before. And all this radical chemistry, again, used to be in the 60s and 70s. It was all about halogenation and studying how this works. They were trying to make refrigerants. They were trying to make various chlorofluorocarbons that could be used for industrial processes. As we discovered this damage to the ozone layer, it's been phased out. We use other molecules nowadays. So it's not as useful industrially, the halogenation, but the antioxidation market, when we discovered just how important that was biologically, it happened to coincide that discovery and that field expanding, exploding, happened to correspond to when we also simultaneously were doing less halogenation. So the free radical industry has transitioned from halogenation years and years ago, generations before, long before us, to polymerization, which was generation pretty much polymers really hit their heyday, just started really doing a lot of polymerization reactions in the 80s and 90s. It continues today. I mean, polymers are very big industry. And then antioxidants, probably closer to the 90s, early 2000s up till now, that's become more of a focal point in terms of research and understanding free radicals. So there's a certain amount of history. We are going to move along that history chain and cover more of the more modern day approaches. So free radical initiators, um, they're oxidizing molecules. They're things that cause oxidation or that cause oxidation typically. They cause free radical formation. So certain molecules readily break apart and turn into free radicals. And as a result, they can do the initiation step. So if I want to carry out a reaction in a certain way, then I will just put the molecules I need to combine with a small amount of initiator, send light into it, and trigger it. There is a chance a good number of you listening to this have actually experienced this firsthand when you went to the dentist. Really, really cool application is the way fillings are done nowadays. They're polymer-based fillings. And so what they do is they put a series of monomers in a solution. It is terrible smelling. Hopefully it doesn't get on your tongue because it's terrible tasting, but they put it into that uh, cavity region, and then they have an initiator in there that's triggered by light. They send light in, so okay, you probably hear the click or you see the glow. It's a high energy blue light, so a lot of times you'll see there's an orange disc of filter to prevent that high intensity light from hitting you or the dentist's eyes. They'll zap that material, initiate it, and it'll start forming polymer and it'll harden. Then they put a slightly different mixture in there, do the same thing, and repeat it and layer this. So they form a polymer that builds up that they can then mold or sand into whatever shape they need. Really cool chemistry. And again, if you're going into chemistry as a career or you're going into dentistry, that is a really interesting one to follow. So material science, polymer mechanism of, I guess, formation, polymer properties, just really wide open field. Very, very interesting field. Okay. Um, these initiating molecules, though, they're critical. They have to be in there in low concentration, but they get it started. And so, for instance, if you want to make a very long polymer, you put very little initiator in there and lots of monomer. If you want shorter ones, then you put more initiator in there so that you initiate more starting material and it runs out faster and gives you a shorter average length. So you can actually customize the nature of the material. Shorter chains are a little bit softer, longer chains get a little bit harder. We know this with waxes, same thing with polymers. And so, again, I'm not well versed in the dentist exact materials they use. But as they keep on doing the layering, the amount of initiator changes with each layer of their build. Okay, so molecules that are very typical. You probably already heard this. Benzoyl peroxide. It is in a lot of cleaning solutions, facial cleaning solutions in particular. But it kills bacteria because it forms a free radical, which reacts very quickly with bacteria. They die off. Problem solved. AIBN is a very common industrial initiator. And then NBS is a very common one that we use in lab to initiate some, I don't know, free radical chemistry or free radical specific reactions in synthesis. So we have a biological cleaning agent that we use industrially, but a biological example, an industrial example, and an OChem synthesis lab example. So I want to focus on this one. If you put light in here, an OO bond is extremely weak and it will break. So let's see what happens. We put the light in. There it goes, zap, 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 forms free radicals. Those free radicals are free to move away from one another. So the benzoyl peroxide has now been cleaved by light. This is highly reactive, although it is resonance stabilized, highly reactive, and it will interact with the bacteria. When it does, it will take a free radical, it'll stabilize itself, turn the bacteria into a free radical, which makes the bacteria literally start reacting with itself and decompose. So I'm oversimplifying it, but it transfers the radical to the bacteria, which can't handle it, and it will break it down. Okay, the way these other ones work, this is going to break, this is going to break. And so if we do that, that's going to be the way things go. Let me go ahead and draw what you end up getting. I'm going to get rid of my arrows. That carbon bond to nitrogen is going to break. That's now gone. This carbon nitrogen bond is going to break. So this one is now gone. And it ends up, you get a third bond here, so you get a very stable molecule, N2, but you end up with two free radicals that are resonance stabilized to the cyano group. So it ends up, you get two very stable initiating molecules, and N2, which is a gas, it's just going to go away. And then the last one, we'll look at this synthetically, but this one, you basically get this. So I'll get rid of those, get rid of the bond between here. You end up with a bromide free radical, and that can resonate over there, that can resonate over there. So it is highly stabilized by this resonance. So a little bit of light breaks this open, and this open, and these can do their biological activity. These can do their industrial polymer initiation activity, and this can do its synthetic activity. Okay, so free radical initiators. I want to look at this one again. So this is going to break this way. You get this free radical. This is initiation. What this can then do is the Br is now free to abstract an H. You end up with that free radical. Propagation, the hydrogen extraction. If you have Br2, this can come in, pick up Br. So you can actually end up brominating something like a, I don't know, cyclohexene at the vinylic position, or allylic. Vinylic, if it's a double bond, it's down the alley, so it's not vinylic, it's allylic, it's one away. So allylic, or you can also do phenylic or benzoic, depending on what you're looking at. Phenylic would be hard to do, but you can put it one away from double bond. Okay, so the point of this being is I can now put a BR selectively next to a double bond by using this initiator. So synthetically, this becomes a really useful combination. So again, let's see what cancels. The Br for radicals will cancel. The free radicals there will cancel, and you're going to end up getting cyclohexene plus Br2 goes to selectively brominated cyclohexene, and HBr is a side product. So you can put a Br alpha or one carbon away, allylic, to a pi bond. So hydrogen abstraction. Free radical polymerization. So that's the synthetic part. You can put a specific leaving group next to, and therefore functionalize next to a pi bond. Absolutely critical type of reaction in a lot of our synthesis. Okay, polystyrene could be poly anything, but for polystyrene, we start with an initiator, have to have it. Initiator is going to come in, ABIN, or ABN could be what we use, who knows? And then we put styrene, which is this benzene ring connected to a two-carbon alkene right here. I hope they line up. This is wishful thinking that they're all going to line up just right. But if they do, the initiator can go in here, form that, and then a free radical's here, which then can find this one, which forms. And so if we're lucky, you get this chain reaction where it adds here, and then it finds another one solution, adds there, finds another, finds another. So the more initiator you put in, the more they're initiated. So you won't have available ones here. So you end up short chain. If I put a really small amount in here, it can only find another, find another, find another, find another. You end up then with this long polymer backbone with functional groups that layer in a certain way. So depending on the properties of this group, these are planar, so it stacks in a very certain way, you get a very rigid polymer. So it's depending on how long it is. But if you get very short chains, then they are free to stack a little bit differently. You get a little bit softer or more malleable polymer. There's so many other features that are going on here, but polymer length is going to have a bearing on it. And then the properties of the side chain and how they interact is also going to have a impact on how they layer, how they pack together. So this is a field that is so booming because designer polymers are critical. You think about our body, polypeptides, aka proteins, they're a polymer. Polysaccharides, polymer. Just DNA, polymer. We're built from different backbones with specific groups coming off. Absolutely critical to our existence. So just like DNA can store lots of information, there's research going on where they have different functional groups here, and polymers can be used to form or store information. So it's a pretty powerful field. Polyvinyl chloride, PVC, you used it in a couple of things. You probably have had a styrofoam cup at some point in life, or a styrofoam cooler, or styrofoam container, and you probably have seen piping for outdoor plumbing, which is made of PVC. So depending on how we cure it, whether we make it fast or slow, whether we bubble air through it or not, we can completely change the property of how it layers. It's the same compound, but again, the initiator to compound ratio will affect the length of the actual strands and the condition in which we cure them in, fast or slow, natural layering or non-natural layering. There's so many factors that come into play. You can literally change the property. So if you think about PVC pipe, that white plastic piping, and then styrofoam, you have to agree they seem like very different compounds. There's actually the same monomer put together in a polymer under different conditions. Does its thing, goes across, forms one to the next, to the next, to the next, depending on the ratio. So you end up with these chains. And again, these chains will layer together and form your overall compound. Okay, so useful radical reactions, that's again modernizing. So polymer chemistry, everything I was doing there is probably its heyday was 80s and early 90s. It's that I guess that type of theoretical chemistry, and today it's a rich, full field. So it came into theoretical existence, then just minimal research existence, and finally a major field. Useful radical reactions. Bio is what I'm going to go to. There's a molecule known as a prostaglandin. And depending on the depth of biology you've studied, you probably have encountered it. It's a messenger molecule that is not glandular, meaning it's formed on demand. So, really good example, if you were to cut yourself, you are exposing internal cells, ones that do not want air to get in because air is destructive, air oxidizes. So they have these molecules that get oxidized by this air. Arachidonate is such a molecule. So uh, upon like some sort of injury where you sever like your skin and expose internal cells, they will release this arachidonate. So lipases will come in, they release it. It will react with the air, two oxygen molecules. It's known as a cyclooxygenase pathway. And in doing so, it'll form a precursor prostaglandin. So it forms PGG2, followed by PGH2. And just point of interest, the A, B, C, D, anything after that. So PG, the first PG here, stands for prostaglandin. G2 means two things. Number one, the two means there's two double bonds outside of the ring system. And it means it was the seventh prostaglandin that they discovered. First one they discovered they called A, second one they discovered called B, third one C. It has absolutely no bearing. It just happens to be arbitrary name. For anybody who's ever worked in a bio lab or a biochemistry lab, it's really common to give a general name to something. It's just what we do. And so those names sort of stuck. So this one, you form this, which then is going to decompose or rearrange and turn into something slightly different. The way this happens, O2, again, keep in mind, is a biradical. Two radical atoms here. They will some way react with here. So this and one of the pi bond electrons, this and one of the pi bond electrons, leaving these two to have to form there, this here, and this here. So I'll get rid of that and see if it correlates. So this O to this one, check. This O to this one, check. And this new bond there, check. So I'm going to end up with that free radical chemistry, which forms that structure. Okay, so that structure is formed. It's autooxidation, does it by itself. It's going to go on to PGH2. So the COX enzymes will kick in, cyclooxygenase enzymes 1 and 2 will kick in, and you end up with this precursor. This can be turned in and modified into pretty much anything here. You can have carbonyls or alcohols. You're pretty much stuck with this one getting oxidized as an OH in most structures. This stereochemistry can vary slightly, but it's very rare that it does. And so some of the ones they discovered. So for instance, you can get PGGE2, which is something that we know signals for pain and swelling. Fun fact, if you take aspirin, it binds that oxygenase enzyme and prevents the formation of the precursors. So you can't get these molecules. If you can't get these molecules, you can't turn them into the pain and swelling. So it's a pain reducer. But unfortunately, it's also a thrombin suppressor. So if you need to clot, which is prostaglandin driven, you can't, or thromboxane driven, but it's the same basic idea. If someone in labor, uterine contraction is triggered by prostaglandin, it's a local response. Prostaglandins are usually local. So if you give them aspirin, you actually take away their ability to experience contractions and you delay the birth. So you cannot give somebody in labor aspirin. Won't work. So this is the one we mentioned, thromboxane. So this was the first thromboxanes we discovered. Notice thromboxane is very similar. The chain is the same here. Instead of a five-membered carbon ring, it's now a five-membered ring with an O inserted there. So these five with an O. So one of the O's is bonded to these two carbons, just like before, but the other O that's bonded here is actually now bonded to the different carbon. So this one inserts in here and this one bridges across. So very similar structure, but it I guess interacts with O slightly differently, but comes from the same pathway, same inherent process to get to here. Okay, you don't have to know these for OChem, but if you do know the features of them, the properties of the double bond, the length of the carbon, the reactivity, the oxidation state, the chirality, it will make you understand your biology much better down the road. But useful free radical chemistry are all prostaglandin, thromboxane, and leukotriene systems. all come from this. So everything that are arachidonate derivatives. That covers the polymerization, and the OChem lab aspect. Hopefully you see the utility in this. It really is an interesting topic, but we are done with free radicals. So in the next podcast, we'll go on to a completely new and wonderful topic. But for now, I'll just stop this one. Thank you for joining, and I'll see you the next podcast. Bye.